Then we are now moving into Chapter 3. If you're not reading or you haven't read Chapters 1 and 2, I would highly suggest that you do that because you'll read Chapter 3 next week and then your test will be on Thursday. So please, if you haven't done that, it's a good idea for you to do that. So the building blocks of humans. We've talked about how DNA goes to RNA, RNA goes to the amino acids become proteins, proteins build up your cells. That's what I like about science, is it makes sense that these things are happening, and I think it's kind of cool the way everything works together. So first, you don't have to write any of this down, but what we're going to be talking about in this chapter is describing what cells are and the two types of cells, the structure and function of the cell membranes, the outside of the cells, the ways in which the molecules get into cells and move out of cells across that membrane, how cells are connected and how they communicate with one another, and then the 9 important landmarks. They call them landmarks. They're the cell organelles in eukaryotic cells. So first and most importantly, what is a cell? All organisms are made of cells. That's part of cell theory. Everything that's alive out there has at least one or more cells, and if you don't have any cells in you, then you are not alive. So if you think about how fascinating that is, think about the fact that every living thing out there, looking at this one, we're just talking about eukaryotic cells, so not bacteria, but everything out there is made of this common denominator, which is a cell, which is really amazing, that this smallest unit of life that can function independently, because cells live on their own, and they can perform these necessary functions of life, including reproducing itself, that they have reproduced into so many different structures. You don't think of your cells being similar to a fungi cell, or being similar to, I'm not sure exactly what that is in the left-hand corner. I have no idea. It looks like a dust mite. All the way up to elephants. It's amazing that all of these things are made of the smallest common denominator. Cells were first discovered by Robert Hooke, who was a British scientist in the mid-1600s. He was, and if I think of the story, he was a monk, and he lived in a monastery. If you think about what a monastery is, it's got all of those little compartments in it, or the chambers where each one of the monks lived. Or if you kind of think of your dorm room, each one has like its own little section, and then you go down the hallway, next section, go down the hallway, next section. So when he looked at a piece of cork, like the stuff that your wine corks are made out of, he looked at a slice of cork under a very rudimentary microscope, and he noticed this three-dimensional structure that looked like a fluid-filled balloon where these chemical reactions were taking place. And so he called them cells after the cells that each one of the monks lived in. And he did not know this at the time, but nearly every cell contains DNA, or deoxyribonucleic acid. And so if you were going to take a look at a cell, most of us think, oh, the first thing I do is get out a microscope. Well, you can actually see cells. Think about the eggs that you eat in the morning. That is one single cell. If you think about how diverse those things are, here we have a hummingbird egg, which is very, very tiny. Oh, excuse me. All the way up to a elephant bird, which is now extinct, that has 2 gallons of fluid on the inside. So cells can be humongous, cells can be little tiny. If you think about how much cells can cost, if you get sturgeon eggs from, or beluga caviar, $700 per ounce. If you think about a human egg, people will pay thousands of dollars to get a woman's human egg if they're incapable of making it on their own. But the problem is getting it out of her alive. It's very difficult to get a human egg out. It involves lots of hormones and very invasive procedures. So we don't just go to our microscope when we're thinking about our cells. Cells go from the largest, which are eggs. They can be the most expensive cells that you get in the entire world. So cell theory, like I said, the first tenet of cell theory is that all living organisms are made up of one or more cells. So if you're not made of cells, you are not alive. And then secondly, that all cells arise from other pre-existing cells. Now, as you might notice, that kind of presents a problem for evolution. One that we have questioned, well, how do we get a living cell from non-living or from the primordial soup? So scientists have been trying to explain that over hundreds and hundreds of years. And there are actually the idea that the cells may have come together from primordial soup. There is a competing theory, which is that cells may be alien life forms that came from another planet. That's not something that we've been able to disprove yet, so it's not off the chopping block. But most likely the first cells came together as this primordial soup. We'll talk about more about that in Chapter 9. So the first type of cells that we're going to talk about today is prokaryotic cells. When you hear that word prokaryote, think of primitive cells. Prokaryote cells are very structurally simple, but there are many, many different types of them. When we talk about cells, every cell on Earth falls into one of two basic categories. It's either a eukaryote or a prokaryote. A eukaryotic cell has these organelles where you've got the nucleus that holds the DNA, and it has things like mitochondria. And a prokaryotic cell does not have the nucleus. It simply has the DNA floating around inside of it. And examples of eukaryotic cells is basically everything besides bacteria. And then prokaryotic cells are bacteria. So here is the prokaryote. Here's a basic bacterial cell. Inside of a prokaryote, it lacks a lot of the structure that eukaryotic cells have. It's got its DNA wound in the center of the cell. It's got cytoplasm where everything's sitting in that jelly-like material. It has ribosomes, which are basically the packaging centers to put together the proteins. It's got DNA here in the center. Cell wall is here, what's around the outside. It's keeping this single-celled organism protected from the outside environment. Sometimes it has both. Sometimes it has one or the other. Pili are hair-like projections. So basically, these are the ways that they communicate or that they stick together. Here you can see a bacteria that has flagellum, and it also has the pili around the outside. So flagellum, same thing that a sperm has, basically whips back and forth to allow it to swim through whatever fluid it is. And these hair-like projections let them stick to surfaces or stick to each other. And so what's interesting about that is that the pili are what causes you to be sick when you get a bacterial infection. Like if you have bacterial pneumonia, those pili are what makes it so that it sticks inside of your lungs. So all of those things are like little Velcro sticking it to whatever makes it sick. And then the flagellum are what makes it be able to swim around and get wherever it is that it wants to go. Eukaryotic cells have these compartments with specialized function. So whereas the prokaryotes are very simple, very primitive, they're lacking in these compartments inside of the cell. The eukaryotic cells have compartments and organelles that allow them to go through specialized functions. So eukaryotic cells have organelles. Here you can see this TEM stands for, I believe it's a telemetric microscope or a telemetric electron microscope. Here we're looking at 6,000 times. Here we're looking at a bacteria that's 10,000 times. So in the typical eukaryotic cell, you have the DNA that's found in the nucleus, which is in the center of the cell. You've got internal structures that are organized into compartments. So if you think back to your Golgi bodies and your endoplasmic reticulum, and they're bigger than prokaryotic cells, about 10 times bigger, and they contain these specialized structures. But the typical prokaryotic cell has no nucleus. Notice its different shape. The eukaryotic cells are shaped one way. Prokaryotic cells, sometimes they're spherical, sometimes they're rods, sometimes they're spirals. But the things that they have in common: no nucleus, they have no compartments, and they are much smaller than eukaryotic cells. So here are the two types of eukaryotic cells that we talk about. We have the basic animal cell, and it's got the nucleus there in the center. So these are the things that they all share. Structures found in both of these types of cells. The nucleus is found in both of those. That's where the DNA or the genetic material is held. We've got our plasma membrane, which is this membrane around the outside. Note that the animal cell only has a plasma membrane, where the plant cell has the plasma membrane on the inside, and it's got a cell wall on the outside, which allows it to keep its structure. It's got ribosomes. Both of them have ribosomes, and the ribosomes, some of them are stuck here to the rough endoplasmic reticulum, and some are floating freely here around the cells. We've got our ribosomes, which are packaging our proteins. Here we have mitochondria. Mitochondria are these little bean-shaped organelles where your cells are going through metabolism and producing lots of their energy. You've got your rough endoplasmic reticulum, which kind of reminds me of like a fruit roll-up. It's going to be kind of wavy and found there around the nucleus. And it's got the ribosomes that are stuck to it. We've got our smooth endoplasmic reticulum, which is kind of reminds me of sea sponges, and it doesn't have any ribosomes attached to it. Cytoplasm is that jelly that the organelles are all sitting in, and there's these little spicules. They are little they look like toothpicks. They're basically making it so that everything doesn't slosh around inside of the cell. They're making it so that the cytoplasm is kind of firm and held in its place. The Golgi apparatus is similar. It's a packaging center. Lysosomes are like a waste treatment plant, getting rid of the cellular wastes. And one of the structures not found in plant cells is called a centriole. And there are three structures that are not found in animal cells but are found in plant cells. And that is the chloroplast, where they go through photosynthesis, make their own food. The cell wall that gives it this rigid structure. And a vacuole, occasionally found in animal cells, but for the most part, vacuoles are just like a swimming pool. They're keeping the water that the plants need inside of them so that plants are nice and crisp and have turgor pressure. So one of the ways that scientists have tried to understand how cells could have come from no cells, how could cells have first started in the beginning, is the presence of two organelles in eukaryotes, the chloroplasts in plants and algae, and mitochondria in animals. And basically it says that those things developed first, and then as those things developed, the chloroplasts making the energy, and mitochondria or chloroplasts making the energy for plants and algae, and mitochondria making the energy for plants and animals, that those things were engulfed in that plasma membrane, and eventually cells formed around those things. So they're trying to explain it, not exactly there yet. So that is why it is hypothesized that at some point humans deep down may actually be part bacteria, because bacteria had those energy-making organelles. They had the components of them first, and then humans, if you think back, we're talking millions and millions of years ago, that those bacteria may have become the origins of our first cells. And that may be why everything out there that's made of cells, it all has DNA, it's all got similar characteristics, because it may have all formed and come from a common ancestor way back there in the past. So in endosymbiosis, this may have been the original cell, where we have ancestral prokaryotes that are converting food and oxygen into energy, and then the ancestral eukaryote takes the prokaryote in. So a prokaryote is the bacterial cell, and so the ancestral cell may have taken in this prokaryote, and when it took it in, it merged, and it may have become the first mitochondria. So organelles may have developed by endosymbiosis. There's also an interesting process called invagination. Yes, it is what you think it is. DNA found surrounded by a plasma membrane, and as it folds in on itself, that's the process called invagination, where it folds in, and it may have eventually become the nucleus and the rough endoplasmic reticulum, and that's how the inner compartments are formed. So we will find out at some point how that happened.